Today, our topic of discussion is jumping gene. Jumping gene was discovered by Barbara. It was discovered by Barbara McClintock, which was an American scientist who, in the year 1950, discovered a mobile genetic element within the core genome that can move from one place to another. This very discovery changed the entire concept of the gene, old gene concept, according to which every gene has a fixed position on the chromosome that is called locus. But she told that gene can change its position, and due to this property of some particular gene, she was unable to map the gene, because mapping of gene is entirely dependent on the fixity of its location, whether the gene is fixed or not. Linkage map cannot prepared for a mobile genetic element. So she told that she named this mobile genetic element she called this mobile genetic element he found there are two kinds of the mobile genetic element, AC and DS. AC stands for activation, activator, and DS stands for dissociation. Dissociation. Actually, you find there are different colors of grain within a cob. This is the cob. Cob is the female inflorescence of the corn. Here you find there are different the grains having different colors. Some grains are yellow, some grains are purple, some grains are purple. Let us pick a different color for it. Some grains are purple and some purple. The whole grain is purple. And some grains show variation in this color pattern. This pattern might be expressed either in the form of patch or spots. She was surprised to get such kind of phenotypes, because the purple color of the grain occurs due to the expression of anthocyanin gene in the aileron layer. Anthocyanin pigment in aileron layer of corn seed. Anthocyanin pigment there is gene for anthocyanin synthesis. The gene for anthocyanin synthesis, the gene is called, the wild type of gene is called C, or sometimes C or C plus. C or C plus. The expression of this gene causes purple coloration of the kernel. Purple coloration of the kernel. ये क्या करता है? Purple coloration. Purple color of kernel. But due to presence of this mobile genetic element, this color is not uniformly expressed in every seed. For example, that there are two kinds of this element, as we have already mentioned it, activator and deactivator. These elements gets inserted into this gene. Suppose this is the kernel, this is the gene for anthocyanin synthesis, normal gene. It will obviously gets translated, transcripted and finally translated and produce anthocyanin pigment. But if these sequences, whether AC or DS, gets inserted into this gene, AC or DS, because these are mobile elements. These can jump from one place to another. Sometimes these get inserted into this gene, functional gene, and making this gene inactive. This way, the color cannot. This pigment is not forming, and due to this, there will be yellow coloration. Yellow coloration will be due to the insertion of this DS element or AC element in the coding sequence of the gene for the anthocyanin. Now, there are various possibilities of this kind insertion. One thing we should know that here there are these two elements, activator and dissociator. Here is the activator, here is the dissociator. Activator and dissociator, these are the transposable element. We have discussed in our last lecture that what are the transposable element. ACDS are the transposable element, specific gene sequences that can change its place from one place to another, either within the chromosome or they can also make interchromosomal movement. Here, the AC activator is a complete gene. It can produce its enzyme transposes, and DS cannot produce. DS no transposes. Actually, this is the enzyme which helps in the movement of this controlling element from one place to another. If there is no transposes, these elements cannot move. Since AC can produce its own enzyme, it is autonomous. It is autonomous transposon. It doesn't require any help from other genes for its movement. But DS, since DS has no transposon, it has no transposes, it cannot produce. Actually, it cannot move on its own. It requires the presence of transposes, the enzyme which can help in its movement. Therefore, DS can only move in presence of AC. DS can only move in presence of AC. Both AC and DS both carry similar sequences, because since they are conservative transposon, we know that the basic structure of the conservative transposon. Here is the basic structure of the conservative transposon. Here we see that there is a coding gene for transposase enzyme, and this coding region is flanked by inverted repeat on either side of this coding sequence. These inverted repeats helps in the removal as well as integration of this particular transposon to removal from the donor side and integration to the recipient side. The detailed mechanism we have discussed, the detailed mechanism how it make cuts on the donor side and make cuts on the recipient side at the time of integration. We have already discussed in our last lecture. Here you will find that this transposase enzyme is not produced by this DS element. Otherwise, both are very similar. Therefore, DS is called deletion product of the deletion product of AC. Deletion product of AC. DS is nothing but deletion product of AC. Both having the similar inverted repeat. And the intervening sequence, that is transposase, is not complete in DS, but it is complete in AC. Due to this lack of a complete sequence, DS cannot produce, and it is non-autonomous. That is, it is non-autonomous. DS requires the presence of AC for its movement. Here is the C gene, C capital and C small. C capital is the dominant gene, and capital C, small c is heterozygous condition. Capital C will give you the purple color, capital C, and capital C, small c. And yellow color will be produced by small c in homozygous condition. But how can you explain this? Here you will find there is tissue specific expression, and there are the expression of C gene is restricted to some patches or stripes, but it is not expressed in the entire kernel. Here we see that C gene gets inactivated. Suppose AC or DS element, either AC or DS elements get inserted. This is producing purple color, functional gene, but now it is called CT gene after insertion. The CT gene is inactive. Since this gene is inactive, it will produce yellow color. We know that after fertilization, seeds, the size of seeds grows due to the rapid division of zygotic cell as well as endosperm. Endosperm cell also keeps on dividing. and finally seed gets its mature site, full site. During this rapid cell division, sometimes this DS elements gets inserted into this, and sometimes it moves out of this. When it gets inserted into this anthocyanin gene, the color stops expressing, pigments stops forming, and this results in yellow color. But when there is continuous to and fro movement of this DS element, resulting in formation of color in some tissues and lack of color in some other tissues, and finally you will find this kind of patch or spots. There are two possibilities. These are the insertion mutation that is done by this AC and DS element. There are one more possibilities that suppose this sequence is the DS sequence, DS, and this one is anthocyanin C. DS gets inserted and it moves out. When gets inserted, then yellow color. When moves out, purple color. The insertion of this movement, DS element, I have already told you, it also requires the presence of SE in the same chromosome or at some other chromosome, because DS is non-autonomous. It requires the presence of AC somewhere else in the genome. It might be present on the same chromosome or it might be present on some other chromosome. AC is producing transposase enzyme, and that is helping in the frequent movement of this element. One more thing is the site of integration of this DS element. Suppose if this is a gene of C, purple anthocyanin gene. If DS element gets inserted here, DS element gets inserted within the coding sequence, then there is possibility that it can move out and finally restoring the color of the endosperm. This very arrangement is not permanent. But if DS element, there are two sides of integration of this DS element. If DS element is attached here, ahead of this, in front of this C gene, it will cause chromosomal breakage. Chromosomal breakage means that here is the chromosome, remaining part of the chromosome, and this very part will be lost because it will introduce a breakage at this point, and after the loss of this segment, what you will find, because the gene is gone now, you will always find yellow color. because now the chromosome fragment is lost, and now this change is permanent. It means that now it cannot gets the permanent, now it cannot gets color due to the loss of this gene is lost. Now with some of the entire discussion, Barbara McClintock discovered this mobile genetic element. It can move and from one place to another. She found that these elements are of two types. She called them activator and dissociation. Activator is a conservative transposon having its own enzyme coding sequence for the enzyme, but DS is the deletion product of AC. Though the sequence DS shows sequence similarity with the AC, but it cannot produce functional enzyme transposase, so it is dependent on the AC for its movement. Here you will find that yellow coloration is due to lack of pigment. Purple coloration is due to the presence of pigment. Continuous expression of this pigment and patch and spots of purple color you will see in certain grains, which is due to the irregular expression of this gene that occurs due to activation and due to deactivation of this color producing gene at some stages or at some stages of the development of the endosperm. Finally, we see that this DS element can integrate or insert itself at two places. It can get inserted within the coding sequence of the pigment, or it can insert itself ahead of it. When it DS gets inserted within this coding sequence, this halts the production of pigments, resulting in loss of pigment, but it can move out of it and the pigmentation and the pigment production is restored. But this way you find that if this moves here, this will produce yellow kernel, and this from here it moves to its original position, you will get a purple color. But DS sometimes also get inserted ahead of this chromosome, in front of this chromosome, or upstream of this chromosome. This integration causes chromosomal breakage, and due to the chromosomal breakage, that part DS along with this pigment producing gene comes out of the chromosome, and this results in the permanent loss of color. Thank you.